Hello, my name is Henrik Thurfjell. I work at the Swedish University of Agricultural Sciences, and today I will talk about lessons from estimating genetic indicators using data from the Red List in Sweden. This was suggested by Hoban et al. In 2020, and our study was published this summer, as can be seen above. First, the Red List is a way of ranking the need for conservation effort among species. It's a way we try to get a result with whatever data we have. A lot of different data is collected for the Red List, and here are some examples. Some can be used for estimated genetic indicators, and some can't. When we're looking at the indicators, we can say that for indicator 1, the proportion of populations within species with an effective population size of more than 500 individuals, we can use the Red List database. For indicator 2, the proportion of populations or range maintained within species, we can also use the Red List database. But for indicator 3, the number of species and populations that are monitored using DNA-based methods, we're using gray literature. If we look at these two diagrams, we can see that for indicator 1 to the left, we have almost complete data for the 4 species groups furthest to the left that are vertebrates. And for indicator 2 to the right, we can see that almost all the data we have is in blue bars, which means they are not maintaining range or population. Otherwise, the field is not filled out. For indicator 3, we can see that, again, the vertebrates are the species groups with the most monitoring. Some conclusions are that the data is suitable but could easily be improved. Just by filling out all the fields in the red listing process, we could make a better genetic indicator. If we were to do genetic indicators at the same time as the red list, this would create a synergy and improve both the accuracy and efficiency of both processes. We also saw that the red list wasn't aligned with genetic indicators perfectly. So genetic indicators are looking more at the population level, while the red list is looking more at the species level. We also found some threatened species in the genetic indicators that were not found by the red list and vice versa, so they complement each other in a way. Overall, we conclude that genetic indicators could be evaluated for a substantial fraction of species in the country, and that genetic indicators have the potential to identify threatened species and populations that are missed by the red list. If you have questions about this, you can email me. Overall, I think that conservation genetics is not now at the point where genetic indicators could start playing a large role and hopefully improve the conservation of both species and populations. Thank you.